Okay, good morning, everyone. Let's get started. For those who celebrate, may the 4th be with you. I noticed, well done. So, welcome to the last class of CS 206, Evolutionary Robotics. It's been a long journey. We are going to wrap things up today, hopefully with something that you find interesting, self-replicating frog bots. Hopefully there will be something there for you to find of interest. Before we get to the fun stuff, let's talk logistics, specifically final project. As promised, I'm going to go through exactly what you are expected to be doing before 11:59 p.m. this coming Monday, which is submit your written report and your oral presentation, which we'll talk about in a moment. The following morning, starting at 7:30 in the morning, again, I don't set the exam times. We will all be here at 7:30 in the morning for a little less than 3 hours. And during that time, you are going to see 62.5-minute videos of evolved robots jumping, trotting, galloping, object manipulation, swarm intelligence, and so on. Okay, so let's talk about the written report and the oral presentation, which again is due in Blackboard by 11:59 p.m. this coming Monday. So if you click on either link for the undergrads or the grad students, it will take you to the same document. Undergrads and graduates, you're all submitting at this point the same materials for the same amount of your final grade. Yeah. Let's start with the written report. This is worth 7.5 of your final grade, and it is due Monday night. You can now find a submission in the final project called written report. There is not a separate submission venue for the grads and the undergrads. Everything is going to Blackboard CS 206 final project written report. You're submitting one PDF document, 4 pages, double-spaced, 12-point font. If it's helpful for ChatGPT to help you write this document, by all means, knock yourself out. Okay. Hopefully, you are in the process of conducting your A/B testing. This 4-page written report, hopefully, if you're making good progress with your A/B testing, it's not going to be a lot of work for you. It's basically just reporting out to us the results of your A/B testing. As we've talked about several times now, what you are trying to do between now and Monday night is collect as much data as possible. Hopefully you have a debugged system that can run in either A mode or B mode. Hopefully you're running multiple independent trials of variant A and variant B. You're then plotting that data in some way. The easiest way is to plot fitness curves, and hopefully you start to see a separation between A and B. As I mentioned a few times already, as long as you have enough data and you do not see a separation between your A fitness curves and your B fitness curves, that's fine. You can try and prove to us that there is no difference. If, for example, in A and B, you have no hidden neurons, and in A you have no hidden neurons, B you have hidden neurons, and you have enough data to show that for evolution, doesn't really matter, you can conclude hidden neurons are not necessary for whatever it is you're evolving your robot to do, whatever physical structure your robot has at that point, whatever environment it's in, and so on, right? What we're not looking for is, I have a couple runs of A and a couple runs of B. They kind of look like they're overlapping. They kind of look like they're not overlapping. I don't know, yeah? So the best way to avoid that situation, collect as much data as possible, yeah? Okay. You'll need to generate some visualizations, as I just mentioned, fitness curves. That's one way to do it. We've seen lots of other kinds of visualizations in this class so far. We've seen footprint graphs, phylogenetic trees, whatever you want to do to try and convince us A is better than B is better than A, or there's no difference between the two, great. The more the better. Okay. So as you're performing your A/B testing this week, you can write up your report as follows. The report should contain 4 sections, 1 page per section for 4 pages. Points will be taken off for poor writing quality. Okay, so write well. Write grammatically and syntactically correctly. The first section should describe your goals. What is it? What was the final project that you tackled in these deliverables? How did you create your A and B variants from that final project starting point? What was the question you tried to answer? I'm evolving a robot for pronking, and I included hidden neurons in variant B. My question is that having hidden neurons, which allows for a nonlinear transformation from sensor values to motor neurons, from sensor to motor values. My question is, I think that will help with pronking, or maybe it won't help. Let's see. Yeah, that's a question. Okay. Second section should describe implementation details. How did you code up your deliverables and the A/B test? What was your strategy? Please describe in words. Please do not just copy and paste code snippets.
So is our description of how we did the A/B, like created the A/B test for the first section, is that just like a high-level explanation?
High-level explanation, I guess that could sort of go into the first section and the second section. So in my code base, in order to make variant A and B, I made 2 separate repos and then changed this part of the code base in repo B. Or I have just one code base and I put an if-else statement in there. The if will take care of A and the else takes care of B. Right? So just sort of walking us verbally through your code base. What changes did you make? You know, go into sufficient detail to fill out about a page. How did you go about doing it? Depending on your final project, some of obviously the implementation is kind of straightforward, but for other projects, how you actually accomplish what we're seeing in your images and what we'll see in your videos for your oral presentation are not so obvious. Please describe it here. You don't need to stick religiously to 1 page per section. If you want to spend a little bit more in one section explaining something and a little bit less in another section, that's okay too. Okay. All right, third section is sort of the most important one here. This is where you're providing the results from your A/B test. First thing is to try and prove to us in this third section that evolution did in fact occur. Yeah, show that an evolved robot did better than a randomly generated robot. We've talked about lots of different ways you could try and prove that in an image. In your oral presentation, you're going to try and prove it with videos. Yeah. An image or video or data at this point. So prove that to us first within one variant. Maybe if variant A is worse than variant B, variant A may be so bad that there is no evolutionary progress. The fitness of evolved robots is not that distinguishable from the fitnesses of the initial random robots from generation 0. Yeah? But hopefully in at least one of these variants, you got evolution to do something. Yeah. Okay. Then, again, this is the result of the A/B test. Show us with data and words and description. For those of you that are comfortable with statistical testing, you can perform a statistical test and report p-values, although that's optional in this class. Show and try and prove to us that one variant did better than the other, or that both produce the same quality of robot. You may want to include fitness curves here, footprint graphs, phylogenetic trees. We saw some phylogenetic trees in the Golem project back when we were talking about crossing the reality gap. Visualizations of how robust evolved robots are to new environments. You may remember when we talked about the minimal cognition experiments with the little Space Invaders robot moving back up forth along the bottom of the screen. They took a lot of those evolved CTRNNs, those evolved controllers, and played back that controllers many, many times in new environments to demonstrate robustness. Yeah, that's something you might also want to include. This is all optional. It's all to, again, try and prove what you got from your A/B testing. Question?
Do you have any policy on the size of figures included on page 3?
Let's see. I have 49-year-old eyeballs, so as long as I can see it and zoom in your PDF and see everything and read it, that's fine. Yeah. Okay. Yeah, I'm not a stickler for font size and all the rest of it. As long as it's generally readable, that's fine. Yeah. So obviously in this third section, which should be about a page, maybe a little bit more of a page if you have less to say about implementation details, a mixture of figures and verbal description. Right? If you just fill the third section just with figures with no captions and we can't figure out what the data means that you're trying to present to us, that's not great. Yeah. Okay. You can also include screenshots of the robot in its virtual environment. So if you've made significant changes to the physical structure of your robot, or you have a robot swarm, or you've placed obstacles in the robot's environment, obviously in those cases, it's helpful to show in addition to tell us what changes you made. Yeah. Okay, in the fourth and final section, you should demonstrate that you've thought carefully about your deliverables and the A/B test. So you're going to sort of go back and tell us the story again. When you first started in on your final project, you were going to evolve a robot that woke up and had artificial general intelligence and took over the human race. That was a little overly ambitious, so you scaled back your project in deliverable 1, and then when you got to deliverable 2, it didn't work, so you had to change it to this. Tell us the story of how you got, where you started from and how you got to your final A/B test. Yeah? Okay. What we'd also like you to demonstrate here is some thoughts about the final project. I know you have other final projects and other things going on in other classes. I know you did not have infinite time and mental resources to dedicate to this. What was surprisingly difficult? What was surprisingly easy about implementing all of this final project? If you had another year to work on this project and you didn't have anything else going on, how might you expand your project? What didn't work? What weren't you happy with? Given all the experience you now have with evolution and robotics and perhaps what you've learned in lectures, how might you try and do whatever you did in your admittedly small A/B final test here? How might you do this on a larger scale? What new features might you want PyroSim to have to achieve this? Again, you're working with PyroSim, which hides a lot of the physics engine details from you. You had a very limited range of sensors and motors and joints to work with. What additional functionality might have been helpful to try and do something on a larger scale? And in this hypothetical expanded project, again, given your experience now with physics engines and evolutionary algorithms, neural networks, robots, what do you think, if you were to do that project, would be relatively easy and which aspects difficult? Yeah. So as you can probably tell from the description of this fourth and final section, what we're looking for is for you to demonstrate to us that you've absorbed a lot of the concepts, but also the practical challenges of doing this kind of robotics. And you're going to demonstrate it to us by describing what you might do at a hypothetical project and what might work and what might prove challenging. Make sense? Fair? Okay. Okay. Let's talk about the oral presentation. Like the written report, it is also worth 7.5% of your final grade. It is also due by 11:59 p.m. this coming Monday. You will now see there is a submission venue in Blackboard under final project. Again, there are not separate submission venues for the graduate students and the undergraduate students. You're all submitting your oral presentation here. Yeah. What are you submitting to Blackboard? You're submitting a URL that points to a public YouTube video. Okay, this video should be as close to 2 minutes and 30 seconds as you can get. If it's less than 220 or more than 240, we're going to remove points. The reason why we're being so strict here is, again, we have 60 of these 2.5-minute videos to get through in a little less than 3 hours. If you do the math, you'll see that we're going to be very tight on time. Yeah? So we don't have time for projects that are going a little bit over. If everybody goes over by 10 or 15 seconds, we're already in trouble. So we're going to keep very close. Please try and keep as close to the 2 minutes and 30 seconds as you can get it. Yeah? Okay, so what is going to be in this 2.5-minute video? Like the report, it's also going to have 4 sections. Part of your grade for the oral presentation will be the clarity of communication. We'll talk about that in a moment. So I would have 4 parts to this video: an introductory part of the video showing us what you implemented. Section 2, some details about implementation. In this, I should have written it down. I will include this in the instructions. In the second part of this video with implementation, if you want to include code snippets, that's okay. But remember, this is a 2.5-minute video. If you put up 30 lines of code, no one's going to be able to read it. You might put up a couple lines to show where you made changes. if implementation is an interesting or non-trivial part of your final project. Yeah. Okay. Most important part here, obviously, is the results section of your video. So in this 2.5 minutes, if the intro and the implementation and the final part are relatively short and the results section is pretty long, that's normal, that's usual. Use most of these 2 minutes and 30 seconds to prove to us, first of all, that you managed to evolve your behavior of interest. Show us 2 videos, a random robot and an evolved robot. And ideally, even without telling us what you evolved the robot to do, we should be able to tell. Yeah. So we're not looking, if it's locomotion, we're not looking for, very fast speed, or if it's object manipulation, we're not looking for perfect grasping and perfect lifting. It's that we can see there is a significant progress towards that behavior beyond what you would expect from a random neural controller controlling the robot. Yeah, that's the bar you're trying to get over here. Okay. You will then want to show a second a second pair of videos comparing the best robot from A and the best robot from B. Show a second pair of videos comparing the best robot from A and the best robot from B. And again, best case scenario here is you have enough data that you see a real clear difference between A and B. Take your best A and your best B, and if you play those 2 videos, again, we should be able to see immediately that there's a difference, right? Depending on what you chose, you may or may not be that lucky. Then you got to fall back on data and visualizations, fitness curves to try and prove to us A is better than B is better than A, or look, I have enough data to conclude there is no difference between these 2. Whatever we provided evolution in B, it didn't need it. It wasn't necessary for evolving whatever you were trying to evolve. Sound good? Okay. Your video will have no sound, so I suggest, especially for a 2.5 minute video, little to no bullet points. If you want to have some very short sentences and maybe 1 or 2 bullets, okay. With my grad students, I outlaw bullet points altogether. Best strategy, since you're using the medium of video, is show rather than tell. Videos, fitness curves, code snippets. The reason why this is all going to be up there with no sound is you're going to be talking over your 2.5 minute video. Yeah? Okay, so during the presentations, when it's your turn to present, you will come to the front of the room and you will talk as your 2.5 minute video plays. Okay. I guess this sort of goes without saying at the moment because you've done a lot of this already. If you don't have any screen capture software, you can record your screen with your phone. Just make sure everything's clear enough that if you play your video on this side screen and you're sitting in the back of the room, people in the back of the room can see what you need, right? So if there's a very small detail, reshoot your video. Okay, if the video is so poor that we cannot see what you're describing or pointing to or explaining during your presentation, we're going to take points off for that. Okay. By 11:59 p.m. this Monday, upload your video to YouTube. Make sure the link is public, because what we're going to do is we're going to stitch all 60 YouTube videos together into a playlist. And at 7:30 a.m. on Tuesday morning, I'm going to press play on the playlist and hopefully not press stop. We're just going to let them all play, and each student is going to come up and talk over their presentation. Yeah? So if you submit a link at 11:59 p.m. Monday night, and our long-suffering TA, Krishna, is going to spend the early morning, Tuesday morning, stitching them all together. If he can't open your video, it's not included in the playlist, and you will be getting a 0 for the oral presentation. Again, apologies for being strict, but we need to make sure everything works smoothly to make sure we can get through all 60 presentations on Tuesday morning. Okay? Okay. All right. Once you have uploaded your video to YouTube, copy and paste the YouTube URL into Blackboard, hit submit, and you're good. You're done for Monday night. Good? Okay. All right, as I just said, Krishna is going to stitch all 60 videos together into a single YouTube playlist. Every year at about 6 in the morning, a student emails us and says, something went wrong, I didn't get to it by now, can you please include it? We can't include it just before the presentations start. Okay, so please, please, please make sure it's up there by 11:59 p.m. Monday night. Okay. As I mentioned, if you're even a few minutes late, by the time Krishna is starting to stitch these playlists together and he gets to your name and he doesn't have a submission on Blackboard or he can't open your video, he's going on to the next student in the list. Okay, that's Monday. Any questions about Monday? Yes?
First, can you show us again how you got to this page?
Oh, I'm sorry. Yep. So if you go to the schedule, as always in the assignment here, there's a link that points to this document. I'll put it up on Blackboard as an announcement as well. I'll post links to this everywhere so you can find it. Second question.
Yeah, and when Krishna's stitching stuff together, is there any order to that? Like, is it alphabetically last name?
Yep, I will get to that when I talk about Tuesday morning in a moment. How the batting order for presentations. Other questions about what's expected by Monday night? Yes?
Can I use PPT format to do the oral presentation?
You can make your slides in PowerPoint or Google Slides. Doesn't matter to me what you make it in, but you're then going to record your presentation. So if you make it in PowerPoint, that's fine. You know, start a stopwatch, start recording your screen, and walk through your slides at the pace that you're going to present it. Remember that you're going to speak orally over your video. So if you have a PowerPoint slide and it's got, you know, this amount of text on it, and you're recording your screen, and you pass by this slide in 10 seconds as you're recording, that means you have 10 seconds to read all this to us Tuesday morning. So another big part of this, especially for a very short presentation, like 2.5 minutes, is pacing. When I give very short presentations, I write out exactly what I'm going to say in the presenter notes and then read that as I walk through the slides. Yeah? And then once you know that, walk through the slides at that pace. Better yet, even say it and just record your screen. Now you have a video, put that up on YouTube, and that's what you're going to say over your 2.5 minute video. Doesn't matter to us how you make the video, just make sure it's 2.5 minutes and that you're able to comfortably talk us through it as it runs. Takes some practice. Other questions? Okay, let's talk about Tuesday. Let's talk about Tuesday morning. Okay, we are all going to meet here at 7:30 in the morning. Again, I know that's painful for some of us. Not my choice. Okay. Right at 7:30 a.m. in the morning, I am going to start playing a YouTube playlist on this laptop, and we're going to watch that playlist. 60 videos is a lot to watch all in one piece. So I am going to put 2 10-minute breaks in the playlist so we can all get up and stretch our legs and then carry on to the next presentations. Okay. The schedule at which everyone presents will be made available in the course schedule beforehand, but just the morning of. In previous years, I put up the schedule days in advance, and people only showed up when their presentation was due. I know you're all very busy next week, but everyone here has put a lot of work into these final projects, and I've seen a lot of these projects already. There's a lot of gems in there. So I want everyone to be here and be respectful of your student colleagues to stay for the duration, right? Even if you present right at the beginning or if you present right at the end. Fair? Okay. All right. Okay, so I'll make the schedule available. I'll link it into the course schedule. You can bring your electronic device to class on Tuesday. That's fine. You can keep an eye on the schedule. When the student before you begins speaking, so you'll have a student will stand here and talk over the slide. So if I'm the one who's presenting before you, You'll see that on the schedule. Please come around and stand there. When it's your turn to present, move from that side of the screen to this side of the screen and start talking. Go sit down. Next student comes up. We are not going to pause the video. So the moment your video comes up, that's your 2 minutes and 30 seconds to start talking. Please make use of all that time. It will go very quickly. Okay. When your video starts, please narrate your video and then reseat yourself. This is a pretty good room acoustically. Obviously, I tend to project my voice. Hopefully, people in the back corner there have no problems hearing me. That is not true for all of you, so please speak up when you're presenting. Okay. In order to keep on time, all the videos will play one after the other, as I just said, with no pause in between. I will build into the YouTube playlist 2 10-minute breaks so we can have a chance to stretch our legs. But this means that when your video starts, you need to start speaking immediately. If you run long, you will be cut off and the next student will immediately start speaking. If the other student doesn't start speaking, I will cut you off. In advance, nothing personal, but again, we've got a lot of videos to get through, so this train is not stopping for anyone. So practice. The good thing about giving a 2.5-minute talk is obviously you can practice it many, many times in a short period of time. I had to give a talk years ago at MIT. It was a 90-second talk. They told us beforehand, when you're on stage, when 90 seconds comes on, we're turning off the lights and turning off the mic. No one is allowed to speak over 90 seconds. Drove the 3.5 hours down to Boston with my wife in the car, and I practiced the 90-second talk the entire way. If you ask my wife today, 10 years later, she could probably recite that talk verbatim, although she'd prefer not to, but there you go. Okay, practice, practice, practice. Okay, Q&A session. Again, you're going to see some really amazing projects. And it would be wonderful if we had time for Q&A for all of you to be able to field questions from your fellow students, but we simply don't have the time. So I encourage you to, if you see a project you like, find that student, ask them questions. How did they actually pull this off? Are they willing to share their code if you're interested during the breaks and after the presentations? Again, you all put a lot of hard work in here, so let's make sure we give everyone the attention and interest that they deserve. Okay. Collegiality, again, this probably goes without saying, but I'll say it anyways. 2 hours and 45 minutes is a long time to sit and watch YouTube videos. I know there's a temptation to mentally tune out during the presentations, especially at this time of year when you got a lot of other demands on your time, and we will all probably be sleep deprived by the time we get to Tuesday morning. I understand. However, again, everyone's put a lot of work into these projects. There's a lot of creative effort that you're going to see here and hopefully learn from. I've done all the talking so far. This is your chance to shine. So please practice collegiality, extend your fellow students, especially those that are presenting, the respect they deserve by watching and engaging with their presentations, right? We're all gonna have our electronic devices in class with us for 2 hours and 45 minutes. Resist as best you can. Okay. Thanks for bearing with me as I read through this together, but I want to make sure that we're all literally and figuratively on the same page. Any questions about oral presentations Tuesday morning? Okay. I know things come up, things happen. If it does, we're going to skip over you and carry on. It's just the way we're going to have to do things. So make sure everything's ready to go well in advance. However you're doing things, make sure everything's backed up. I inevitably get emails Monday night that your laptop broke or something happened. So do things in advance, have backups, put things in the cloud, give things on a flash drive to your friend, whatever you need to do so that there's no hiccups Monday night and Tuesday morning. Okay? Okay. All right, I look forward to seeing your presentations on Tuesday morning. Okay, as I mentioned, we are at the end of the course. We're going to spend a few minutes talking about self-replicating Xenobots. I am not going to go through the entire slide deck. I will give you an overview of this final project in evolutionary robotics that we're going to talk about. And I'm going to leave about half an hour for open discussion. We've covered a lot of ground. I promised you we would talk about the ethics of robotics, and I've been putting it off. We can talk about everything you've heard. We've talked mostly about the how. How can you go about creating intelligent machines that may someday work alongside humans in the real world? Let's spend a little bit of time talking about whether we actually want to do that. As roboticists, we can look at our fellow researchers in the field of AI who have been very successful and are now grappling with whether or not we should have created AI. A lot of us feel it seems a little bit too late to be having that discussion because the genie is already out of the bottle. Luckily for robotics, we haven't distributed robots over the surface of the earth yet. We have time to think about whether this is a technology we actually want to try and build, scale, make useful, and so on. All right? Okay, so back to the world of Xenobots for a moment. As I mentioned, we're going to talk about self-replicating Xenobots, and whenever I present on this topic, the first question I inevitably get is, why the heck would you want to make self-replicating robots? Hasn't Hollywood shown us enough what happens if you actually manage to do so? Yes, and we'll come back to that in a moment. So let me preempt that by explaining why this topic is of interest to me and many others in the field. Many of the large-scale challenges that we face at the moment, like grappling with climate change, pulling microplastics out of the ocean, pulling carbon dioxide out of the atmosphere, these are big, big tasks, global tasks. In order to grapple with them or solve them, we're going to have to create technologies not just that pull CO2 out of the air or microplastics out of the ocean. They're going to have to do it at scale. We're going to have billions of devices or machines or processes that are doing this, and they have to be cost effective. For a lot of climate change mitigation strategies, the technological challenges are already solved. We know how to do it. It's the economics that is not solved. We don't know how to do it cheaply enough that someone or some nation state might actually pony up a check to do it. Big challenge. Okay, so how do we scale technologies? There are different ways. One of the original ideas, which has remained an idea and not a practical reality for many, many decades, is self-replication. Yeah? We have a traditional factory, and that factory produces cars. If you want to produce twice as many cars, you need to make twice as many factories. You need to pay for the factory. You need to pay the workers for the factory. If you want to make 10 times as many cars, you need to put in a 10 times amount the investment to make 10 factories, and so on. Yeah? That's how traditional technologies or generative technologies like factories that themselves are not useful, but they generate useful devices or things or processes, works. Wouldn't it be nice if we could make machines that make copies of themselves, and eventually those copies of copies of copies sometimes also spit out cars? If we could create such a system from an economics point of view, we need to make an additional investment to figure out not just how to make a factory, but make a self-replicating factory, and that's also more expensive. But once we figure that out, the cost for the child factories and the grandchild factories is marginal. It's perhaps just the materials themselves, or if the factory is mobile and can move around and find the raw materials with which to make a factory, now you don't have to pay for the materials either. The cost of the second, third, and nth generation of the self-replicating machines approaches zero. So if you want to make a very, very, very large number of cars, you can do it this way, or potentially in the future, you could do it this way. Yeah? So you reduce literal cost, financial cost, through self-replication, but there's no free lunch. There's always a cost. What's the cost of this approach to manufacturing cars compared to this approach to manufacturing cars? assuming you can figure out how to do this, it's cheaper, but there's always a cost.
That it takes longer to start actually making cars?
It takes longer, right? There's a long lead up in which factories are mostly making factories and not making cars. Absolutely. So there's a time penalty here as well. What else?
There's a lot more risk. Like this could go wrong, like really bad. Like if something's like done wrong, then it could like go wrong on scale.
Absolutely. So this is an exponential technology, right? Assuming this machine, whatever it is really good at making 2 copies of itself, and those 2 copies are really good at making 4 copies, you've entered exponential technology territory, which is where Hollywood loves to play, right? So there is also the risk of something going wrong. You've seen in this course many, many times now perverse instantiation, right? Where the machine is doing what we want it to do, but in retrospect, it's doing it in a way that maybe we wouldn't have wanted it to do looking back. Yeah. Again, the AI community is now grappling with this in full public view. Tricky, right? So again, whether or not we would want to do that, there's an ethical discussion there. But from a purely economic argument, this is the way to go, especially if we want to produce very, very large numbers of cars or electric cars or CO2 scrubbers or robots that can track down and remove microplastics or whatever it is we want to do on a large scale. It's probably going to have to be some version of this to scale well. Okay, we've been talking about cars and factories, but this idea obviously applies very nicely to robots, and again, self-replicating robots. Hollywood has had a lot of fun with this idea, but Hollywood wasn't there first. The original idea for this actually originated in the late 1940s from John von Neumann. For all the computer scientists here, hopefully that name is very familiar. All of the computers that we have today are based on a von Neumann architecture. Von Neumann machines is sort of the nickname for any class of machine or robot that is able to make copies of itself, like you see in the cartoon here. You can imagine 2 different kinds of machine replicators. There are machine replicators that's all they do. They just replicate copies of themselves, and it's hard to see how that technology would ever be useful for anything. Probably just dangerous and not a good idea. There's a second type of self-replicating machines that they also self-replicate, but as a side effect of that replication process, they also produce something useful, like electric cars or CO2 scrubbers or solar panels, whatever it is. If you can do that, if you can create von Neumann machines that replicate exponentially and generate or build useful devices or products or services, you have what's known as exponential utility. We've touched on a lot of topics in this course. We're ending with economics here, right? Utility, things that are useful to humans. If you can pull off this technology, you start to get exponentially more of these utilitarian or these useful things. Solar panels, CO2 scrubbers, whatever it is. Yeah? Okay. All right, so this has been an idea for a long time. It's not a reality yet. It's really hard to do. There are a lot of roboticists that are working on things that look a fair bit actually like this cartoon. So traditional rigid robots with jointed limbs that are able to move around and find or retrieve parts and then put those parts together to build copies of themselves. And if you're interested in that topic, I've linked to a lecture that we didn't have time for, which is specifically on this topic of robot self-replication. Yeah? Okay. There's work in this area. It's been slow, it's tricky, it's difficult. So back from robotics to the Xenobots for a moment. Last time, we ended by talking about these little Xenobots, and just as a reminder, these are millimeter-sized machines built from genetically unmodified frog skin cells and frog myocardial tissue. heart muscle tissue. Yeah. So you can see about a dozen of these Xenobots in a petri dish. We're looking down through a microscope. We're looking down onto the top of the petri dish. And Doug Blackiston, our microsurgeon, has sprinkled very, very small red glass beads into the dish. And you can see just through the collective motion of these Xenobots, they happen to be pushing these glass beads into piles. This was the sort of end of that first study, which we published in January 2020. Around that time, we started to think about what would happen if you replaced the small glass beads with more frog skin and heart muscle cells. Yeah? Presumably, those obviously individual cells are smaller than the Xenobots themselves. They would start to push those dissociated cells into piles. And a Xenobot is basically a pile of frog cells. Yeah? So I don't know whether this was sort of a subconscious thing that we had going on, if we had von Neumann machines in the back of our mind, but we were sort of thinking about what would happen if you tried this. And as always, we wouldn't be talking about this if we didn't try it and get something interesting. This is what we got. This is the third Xenobot paper. We're not going to talk about the second one. What's happened between the first Xenobot paper and the third Xenobot paper is at this point in December 2021, when we published this paper, we were now working only with frog skin cells. We are now not going to build Xenobots with the myocardiac tissue anymore. The reason why is frog skin cells have very, very small hairs on their outer surface called cilia. And these very small hairs, they're almost like tiny little tails. They're flexible and they whip back and forth. Frog skin cells, when they're on the surface of a frog or when they're on the surface of a Xenobot, these cilia are immersed in, most of the time, room temperature fresh water. They start to act like very small oars. They're agitating the fluid around the cell. In a normal adult frog, those cilia are beating and they're basically washing dirt and grime and viruses and all that bad stuff off of the skin of the frog. It's a cleaning process. Yeah? You also have cilia in your esophagus, in your airways. When you're breathing in dirty air, your cilia are actually the mucus inside your esophagus is capturing all that particulate matter, and then your cilia are moving that mucus away. You either cough it out or get rid of it in other ways. Yeah? It's basically a cleaning process. What Doug found out in paper 2, which we're not going to talk about, is that if you build Xenobots just with frog skin cells, you get obviously something like more or less a ball of cells. And if the Xenobot is small enough, it's made up of a few enough cells, it's light enough that all of the cilia start to beat and the Xenobots actually swim around in the dish. So what I showed you on Tuesday were walking Xenobots that use myocardiac tissue to walk along the bottom of the petri dish, like walking along the bottom of a swimming pool. All of the Xenobots you're going to see over the next few minutes, these are swimming Xenobots. They're basically moving with using the cilia on their surfaces. So far, so good? Okay. So when Doug started to play around with replacing the glass beads with individual cells, he did the following. He made some Xenobots. You can see some of them here in panel A. These are not AI-designed Xenobots. No evolutionary algorithm here. He just took these dissociated cells and left them alone. What do dissociated cells like to do when they're left to their own devices? As you saw in one of the videos last time, they pull together and form up into a ball. And you can see a bunch of them here. He then took those Xenobots and in panel B here, dropped them into a petri dish that was sprinkled with a whole bunch of individual frog cells. So all the white circles that you see in panel B here, these are dissociated frog skin cells. And you can see the Xenobots are already starting to move and swim around, and they're pushing the cells away. I should mention, I'm going to mention these Xenobots as swimming. They're swimming, but they're not suspended in the fluid. They're sort of touching the bottom of the dish a little bit. So it's almost like you're swimming along the bottom of the pool, and your body is keeping very light contact with the bottom of the pool. Yeah. The individual cells have settled to the bottom of the pool, so as you or the Xenobot is swimming around, it's pushing these cells out of the way. Okay. After letting these Xenobots do this for 24 hours, you start to see that some of these piles of dissociated cells that have formed, those piles start to deform up into spheres. Dissociated cells they're very social. They don't like to be on their own. So when they're pushed into a pile, they reach out and grab a hold of their neighbors and pull inward. And in this particular experiment in panel C here, you can see O, 6 O's here. These are about to become the 6 offspring Xenobots. You'll notice these are the bigger piles here. The 3 P's are the quote-unquote parent Xenobots. At this point, Doug, after 24 hours, takes the biggest of these piles, which are now no longer piles. They're big spherical bunches of frog skin cells. Takes them out of the dish, sets them aside for a little bit. You can see there's still some dissociated cells here. This is just a little bit of a mess. And lets those offspring, those spheres, sit in the dish for about 5 days. During this 5-day period, they're motionless. They're just sitting there not really doing anything except growing cilia. Sometimes that's it. That's the end of the experiment. Nothing more happens. 6 days, 7 days, 8 days, 9 days, 10 days, nothing happens. And these new balls of frog cells just start to rot and die away. So with probability alpha, where alpha is somewhere between 0 and 1, that's what happens. With probability 1 minus alpha, and we'll talk about alpha in a moment, what alpha actually is, some of the time, some of these piles then start to move spontaneously on their own after 5 days. They're big enough, they have enough cells where they've started to grow enough cilia, and all of those cilia are beating, that for some reason that we still don't understand, there's enough of those cilia and they all start beating to exert enough force against the fresh water that this pile starts to move in its own right. Okay. What do you do once you have these moving piles? You put them back in another petri dish. In panel F here, sprinkle more dissociated frog cells, and those offspring, those child Xenobots, start to push those dissociated cells into piles. Like you see here, take those piles out of the dish, and in some cases, around and around and around you go. That's self-replicating Xenobots. Questions? What's alpha here? So alpha is the probability that this process runs out of gas. A common question that comes up at this point is, so how many times around do you go? Doug makes I think it was 12 in this experiment, makes about 12 to start with and starts this process. He did it 4 or 5 times. He started independently 4 or 5 times, and in, I think, 3 of those cases, nothing happened. He got here, 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 here. None of these piles moved. They rotted away. That was it. No replication happened. In the 4th of the 5th trials, the parent Xenobots contracted a fungal infection and died early. No one saw that one coming. In the 5th experiment, the Xenobots went around once. These parent Xenobots, which were made by a human, made by Doug the microsurgeon, they produced big enough piles that those piles grew enough cilia that they started to move. He took those child Xenobots, let them mature, push other cells around. They made some very, very small piles. So these were candidate grandchildren. Took those small piles, put them in a dish on their own, let them mature for 5 days. None of them started moving. That was the end. So in those 5 trials, in one of the 5 trials, Doug managed 1 round of self-replication before this process ran out of gas. Yeah? At the time, it was kind of underwhelming, but then sort of pulling back. No one had really seen this before, so we were still happy about it. What do you think the next thing is we did in this study? Unlike what Stephen Colbert said, we did not carpet bomb the lab. We kept going. We're starting with a bunch of spheres. And it turns out that in order to make good children, they need to be big. You need to have enough cells that they grow cilia, that there's enough cilia on their surface so that they start moving. Small piles, they might grow some cilia, but they don't have enough momentum with the cilia to move, and that's it. So what's next? Change the shape of them. Maybe instead of starting with a bunch of spherical ancestors, maybe if we start with a different shape, they'll be able to make bigger children, more and bigger children, so that we'll have a large number of big moving children, which will be large and big enough to make another round of big piles, and maybe we can achieve more than 1 round of replication. This study, like the first study, got a lot of media attention, so some of you may already know the answer to this question, but what? What is the shape that's better than spherical? Again, we wouldn't be talking about this if there wasn't a role for an evolutionary algorithm here. Any ideas? You want to maximize the surface area of the children, possibly. Remember that we're creating these things, which are then going to try and push and make big piles on their own. Maybe a cube. So luckily we don't have any more snow anymore. But when you were shoveling, you want to go out and push large amounts of small things. What's the shape? A crescent shape, right? You basically want these things to be a shovel, right? You want something that has a very large forward-facing concave mouth to push all of the cells.
If you do that though, won't like the children just end up still being spheres?
So as you're going to see in a moment, we used an evolutionary algorithm to try and find a shape that would produce the largest possible children, and the children themselves do not have the same shape as the parents. They're different. The children so far that we've been able to achieve, they're always spherical. We haven't been able to find a shape for parents where the parents produce children that have the same shape as the parents. The best we can do is find good shapes for parents that produce as large children as possible. Large children with high surface area to volume ratio would probably actually be even better, but we haven't been able to figure that out yet. So when Doug showed us this was before we had brought in the evolutionary algorithm to find the shape. We sat down with both groups. Our group here at UVM, the group at Tufts, said, okay, before we run the evolutionary algorithm, what? What is the best shape we could think of that would make large spherical shapes, large spherical children? And in retrospect, we probably should have realized that what you want is a shovel. You want something with a large forward-facing concavity. None of us came up with that at the time. So we brought in our evolutionary algorithm, and this is the shape it came up with, which I guess rightly so the media referred to as Pac-Man Xenobots. So what you're watching at this point is, let me back up for a moment. You're watching the process between B and C here. So you can see a lot of dissociated cells in the dish. But at this point, it's already started to push some of these cells together. And at this point, it's made these 3 or 4 larger clumps of frog skin cells. And these larger clumps are being pushed together into yet one more larger clump. It turns out that it's relatively easy for these dissociated cells to be pushed up into these balls. At this stage, these frog skin cells are themselves very sticky. So imagine you took some masking tape and you pulled out a whole bunch of tape and you just randomly made it into a ball, threw it into a big dish, made another one, and you threw into a very large petri dish or distributed over the floor a large bunch of sticky balls, and then you came around and had something like a Roomba vacuum cleaner that was randomly moving around. It would push those sticky balls of tape into larger and larger balls of sticky tape. That's more or less what's going on here. One added detail, obviously, here, as I mentioned several times already, is these cells are also helping. They are pulling stuff in. So the individual cells themselves are helping with this aggregation process. Yeah. A few hours later, oops, am I missing one? Okay, all right. So let's talk about how we evolved this process, and then we'll stop there for today. Okay. So how does this work? As always, we're going to run an evolutionary algorithm. What you're looking at is one individual from the initial random population. What is that quote unquote individual? That individual contains 1, 2, 3, 4, 5, 6, 7, 8, 9 shapes. You remember back to our virtual lions and gazelles, where we were evolving populations of virtual lions. Here we are evolving populations of individual Xenobots. In this case, all 9 shapes happen to be spheres. So we take that swarm out of the initial population of random swarms. We've got a whole bunch of swarms. We take this particular swarm, we drop it into the Voxcraft simulator. We sprinkle in a whole bunch of virtual sticky frog cells, which are the green voxels. And you can see that this swarm is making piles. We worked with Doug to figure out what was the minimum number of cells that were needed for a pile to eventually mature and become motile, start moving. I forget what that number actually is, but in this video, none of these piles had enough cells in them that Doug thought they were going to become motile. So we built that into the simulation. It was just a threshold number. But one of these piles, this one here, is 74% towards the size needed to become motile. So we assign a fitness value to this swarm, to these 9 Xenobots, of 0.74. They made one pile. The biggest pile was 74% of the way to becoming a motile child Xenobot in its own right. So far, so good. So imagine we then simulate all the remaining random swarms in the population, and we're assigning scores to them. As you can probably imagine, through random motion, none of them are making a pile large enough to become a child Xenobot. So the fitness values we're getting in this initial population are 0.03, 0.18, 0.74, 0.22, and so on. Delete the swarms with low fitness values and make randomly modified copies. Make randomly modified copies of the surviving swarms. And after a few generations, we saw appearing in the population this particular swarm that was made up of 9 donuts. And these 9 donuts, through random action, end up making one pile that was above the size threshold. that was big enough to become motile. You'll see it again once this, I'll start this video from the beginning. If you keep an eye around here, these Xenobots are making a large enough pile here that's above the size threshold, and this pile is now placed in a new dish with new cells, and it produces a pile. I'm sorry, this pile should not be motile. There's an error in this video. So at this point, this pile is 51% of the way to being big enough to become motile in its own right. So we have these 9 ancestral Xenobots that collectively produce one pile that becomes a child. So it gets 1 point. And then that one child produces a pile that's 51% of the way to becoming a grandchild. So it gets that swarm gets an additional 0.51 fitness points. So this swarm gets a higher fitness value. Why did this swarm do better than that swarm? Hopefully you can tell from this little graphic here, the only thing we're evolving here is shape, as was mentioned earlier. Why?
You have a hole in the middle, then that just makes it bigger.
The hole makes the parents are not actually, the purple ones here, they're not bigger than the spheres. It's not that they're bigger. Yeah, good point. So real Xenobots that have a hole in the middle, again, cells don't like that. They don't like to have too many neighbors. So they will sort of, all the cells will deform and sort of try and fill in the hole. That's a good observation, but it doesn't explain why this swarm of donuts manages to produce bigger piles than the swarm of spheres. This is not a trick question. This is not thinking about thinking is misleading.
Because the donut would have more surface area on the ground, so it can pick up more.
Good point. Maybe the donuts would have more surface area on the ground. We don't know. It's very hard to image the ventral or the bottom part of a donut. Maybe there's an easier explanation.
Is it lighter so it can move faster?
It is lighter, right? There's less material because you're missing some in the center. Watch the donuts themselves for a moment. It's a little difficult to see because this is quite sped up.
I mean, it looks like they're all spinning like they're doing donuts.
They're doing donuts. Good point. They're donuts doing donuts. The spheres also spin.
Do they have more cilia?
They probably have more cilia, which might mean that they move faster. In this simulation, I don't think we captured that. I think the virtual donuts move at the same speed as the virtual spheres. All good ideas.
They seem to be capturing some of the orphaned cells within themselves.
That's also true. You'll notice they happen to sort of bump up and over and some get stuck inside. Also interesting, but not why they make bigger piles. If you're a donut made of soft, pliable material and you're like a Roomba driving around and you smash into individual cells head on, what happens to you, the donut? That doesn't happen so much to a sphere made from the same material.
It sort of like compress into the hole.
That's it, right? So the front part of the donut gets pushed inward a little bit by the cells, at least more than a solid sphere was, which means your leading edge is more concave, which means you collect more cells, right? We weren't that smart going into this experiment. Again, the evolutionary algorithm discovered this and then kept going, eventually discovered this shape, which always has a leading concavity. In this particular case, when this video repeats, you're going to see the 9 parents. They're going to make 3 piles that are large enough to become child Xenobots, which in turn make 1 grandchild Xenobot. Okay, here we go again. 9 grandparents make 3 children. One of those 3 children makes 1 grandchild. Yeah? So we achieved 2 rounds of self-replication. So this particular swarm gets 2 fitness points. And the grandchild actually makes a pile I think there's a mistake in this video as well. In the actual simulation of this swarm, that one grandchild makes a pile that's 81% of the way to making one great grandchild. So a fitness score of 2.81. So what is this evolutionary algorithm doing? Or what is the fitness function selecting for? It's selecting for the postponement of loss of replicative ability. As I showed you when I showed you the physical self-replicating bots, the spheres will make children sometimes. They're capable of self-replication, but rarely. And even if they do, that ability to self-replicate is almost immediately lost again. So here we brought in the evolutionary algorithm to say, there's the hint of a useful behavior here, self-replication. Can we evolve these things to enrich, exaggerate, prolong that desired behavior, which is self-replication? Questions? Okay, as I already mentioned, and you can imagine when we published this, the media were interested in self-replicating frog bots funded by the US military. First question we got from every single reporter we talked to is, why? Why would you do this? Again, going back to the start of this lecture, creating machines that self-replicate could be useful if we can control them because they might be capable of exponential utility. From the late 1940s, when John von Neumann first came up with this idea, the idea since then has been, we'll eventually do this with robots made of metals and plastics and ceramics and batteries and sensors and motors. For us, this was a surprise that we might actually be able to make self-replicating machines, but maybe the first self-replicating machines won't be made from traditional robotics materials. They'll be made from biological materials. Which also sounds a little bit sort of intuitive. Living systems reproduce all the time, so it makes sense that they're able to do it here. The twist is to realize that this is what's known as kinematic self-replication. There are lots of different ways that organisms propagate themselves. Mammals grow copies of themselves inside themselves. Trees grow copies of themselves in the form of seeds on the outsides of themselves. It's all growth and then let that thing loose into the environment. This is not growth-based reproduction. It's move around and find material out there that's already there and combine it or push it together. Kinematic means movement. So move yourself and then move existing raw materials out there in the environment to build a copy of yourself. That's kinematic self-replication. As far as we know, that particular behavior, kinematic self-replication, has never been observed in nature before. All organisms, plants and animals and protists, they all make copies of themselves by growing and then budding off something in some way. So we are not exploiting an existing ability like reproduction to make self-reproducing machines. We made non-self-reproducing machines that then discovered this new way of making copies of themselves. So kind of interesting from a biological perspective as well. Whether these machines will ever be useful or whether they will ever take over the earth and turn everything into gray goo, who knows? We'll see. The future is going to be interesting. Okay, on that note, I will stop talking. We have, I'm sorry, only 6 minutes left. Let's open the floor. Question, we've covered a lot of ground in this class. Things you found interesting, ethical things you wanted to discuss.
I have a question about what we were just talking about.
Sure.
Is their size too big where like they weren't able to move?
Yes, yeah. I've simplified the story greatly for today, but yes, it's possible to make a pile that's too big and then it can't move because it's too heavy and there's too much contact with the surface of the petri dish.
So
Yep.
you kind of like asked the question to make like two that almost form like a mold that could then shape the offspring to be a certain shape?
Good question. So I skipped over one detail when we were talking about this. Where were we? These little inset cartoons here, these actually represent how the parents produce the next generation. So you'll notice in this particular solution, we have these 9 parents, and parent 3, 4, 5, 6, and 7. These 5 parents all contributed 1 or more cells to this pile that became motile. Yeah. So you also have this collaborative or teamwork behavior. This other child only has a single parent. There was only one of these. So it's complicated. It's not 2 parents produce 1 offspring. It's multiple parents can collectively contribute to kinematically self-replicating a xenobot. Yeah, good question. Okay. Other questions or comments, ideas about xenobots themselves? Other things?
How do you deal with this new issue of xenobots that can get infected? And if you have a large batch of xenobots that then can develop a fungal infection, what happens now that you're in a field that exposes you to the problems that most biological creatures experience with disease? Yeah,
good question. The short answer is I have no idea. But again, when we're dealing with things, exponential technology, if you have attrition, a certain fraction of the machines break down for whatever reason, maybe it doesn't matter. All that's got to happen is the alpha, the rate at which more of them are made, is greater than the rate at which they die off from fungal infection, or in the case of xenobots, they die of old age after about 7 to 10 days. It's also, in a way, a good thing that all these bad things can happen, because when you study self-replicating machines, one of the most important things is to make sure you always have the big red button, that these things are not super capable, that there's lots and lots and lots of different ways in which you could repress or stamp out the replication. And as Hollywood likes to remind us, there's no perfect red button. Life always finds a way.
Yeah, so I was just gonna say, like, what's the plan? What would be the plan for like making something like this like work in the real world? Because like in a real environment, you're not like in a field of frog cells. Like,
Yes, of course. So another scary thing about self-replicating machines is when they go out and try and find raw materials. What are valid raw materials and what aren't valid raw materials? We have not even started to investigate that here. So again, hopefully it's obvious this is not something we're going to release into the wild anytime soon. It's basically a test bed to understand exponential technologies and exponential systems, right? As we all learned in 2020 and 21 and 2022, humans are not very good at dealing with things that replicate exponentially. Aside from looking at this through a robotics lens, as maybe this being a potentially useful machine someday or a type of machine, it's also a test bed for understanding whether we can bring in AI, not necessarily to accelerate or enrich self-replication, but snuff it out. Imagine designing a swarm that is designed to go out and try and stop some other system from self-replicating. Yeah? One of the hardest things, obviously, about stamping out COVID is all of the technologies we deployed, like the vaccine, the way in which we made enough vaccine was not through a self-replicative process. It was obviously crazily expensive to try and inoculate the global population, and we're still at it. Yeah? So whether we do it or not, we are surrounded by self-replicating systems all the time. Maybe a potential use for AI would be to create other self-replicating systems that can go out and stamp them out, which raises the inevitable question of then how do you stamp out the system that's trying to stamp out some other self-replicating system? It gets complicated quickly. Yes?
Did the parents in that experience move randomly, or how did you implement the real world those cells moving to the virtual world?
Yeah, good question. So I didn't talk too much about sim to real today. I didn't have time. Do they move randomly? Yes or no? Yes, in the sense of we can't predict how they're going to move, but no in the sense that they tend to always move in these spirals. So you get lots of different kinds of spirals. So in the simulation, we're actually simulating small cilia on the voxels that are applying impulse forces against the cells. So our motors in the simulation are these cilia. We are probably modeling these cilia terribly. Biologists are still learning about how these cilia actually work in frogs and in humans, and we're trying to build in details to our model to more accurately model how groups of cells that grow cilia will swim, how they will move. Okay, we are out of time. Thank you for taking this class. It's been a lot of fun. I hope it's been fun for you too. You have your final quiz due tonight, last one. And then we will see you at 7:30 a.m. back here Tuesday morning. Thanks very much.